Welcome to Creative Peptides. Today, we talk about the solid phase synthesis. Solid phase synthesis means synthesis on a solid support. It is a technology that dates back to the early 1960s. R. B. Merrifield, an immunologist working in the laboratory of D.W. Woolley at the Rockefeller Institute in New York, was required to synthesize analogs of biologically active peptides. In the process, Merrifield perceived that the approach of the day, made up of successive coupling and deprotection reactions in solution, followed by extractions at each step to eliminate unconsumed reactants and secondary products, was very labor-intensive and repetitive, and he concluded that there was a need for a rapid and automatic method for the synthesis of peptides. He suggested that the synthesis be carried out with the first residue attached to an insoluble support, so that purification could be achieved by simple filtration instead of extraction. According to his proposal, a protected amino acid is anchored to an insoluble functionalized support by a bond that resists all chemistries employed during assembly of the peptide. The amino group is deprotected, and additional residues are introduced successively. Each reaction is followed by filtration, which removes unconsumed reactants and secondary products that are dissolved in the solvent. Final deprotection detaches the peptide from the support. 3 advantages were envisioned by Merrifield: high yields achieved by forcing the reactions to completion, less manipulation and consequently less time, and minimized losses of material because reactions and purification would take place without removing the peptide support from the reaction vessel. A typical protocol for the synthesis of a 14-mer on polystyrene resin using box/BZL chemistry appears in the figure. The peptide resin is suspended in dichloromethane, then methanol, and then dichloromethane. The latter swells the resin beads, and the more polar methanol shrinks the beads. Alternating swelling and shrinking serves to expose reacting sites. Methanol is also used to remove reagents that are presented in dimethylformamide because it is miscible with the 2 other solvents. There are several features that are characteristic of solid phase synthesis. There is no loss of material accompanying the synthetic steps because the peptide is never taken out of the vessel. Secondary products issuing from the reagents and protectors do not affect the purity of the peptide because they are quantitatively removed after each reaction. Reactions can be encouraged to go to completion by the use of an excess of reagents. The question of the solubility of the peptide does not arise as it does for syntheses carried out in solution. No purification of intermediates is effected during the synthesis, and operations are repetitive and have been automated. However, monitoring the course of reactions is not straightforward, though some automated systems incorporate measurement devices, and synthetic impurities resemble the target molecule, which makes them difficult to remove at the end of the synthesis. The requirements for solid phase synthesis are diverse. The support must be insoluble, in the form of beads of sufficient size to allow quick removal of solvent by filtration, and stable to agitation and inert to all the chemistry and solvents employed. For continuous flow systems, the beads also must be non-compressible. Reactions with functional groups on beads imply reaction on the inside of the beads as well as on the surface. Thus, it is imperative that there be easy diffusion of reagents inside the swollen beads and that the reaction sites be accessible. Accessibility is facilitated by a polymer matrix that is not dense and not highly functionalized. A matrix of defined constitution allows for better control of the chemistry. Easier reaction is favored by a spacer that separates the matrix from the reaction sites. Coupling requires an environment of intermediate polarity such as that provided by dichloromethane or dimethylformamide. Benzene is unsuitable as solvent. Solid phase synthesis is a technology by which the synthesis of a peptide is simplified. The chemistry for synthesis on a solid support is the same as that for synthesis in solution, except that the protector of the carboxy terminus is linked to an insoluble support, either directly or indirectly. Peptides that are to be cyclized may be anchored through the functional group of a side chain. Chain assembly is by addition of single residues, which is the approach also employed for synthesis in solution. The nature of the appendage on the carboxy terminus provides the unique difference. The appendage consists of a polymer matrix attached to a protector. The nature of the linkage between the protector and the first residue, as well as the method employed to anchor the first residue, is dictated by the nature of the target peptide. The stability that is required of the anchoring bond depends on the conditions that are employed to remove the protectors on the amino terminus of the growing peptide chain, or vice versa. The consequence of the above is that the choice of one option limits the choices for the other options because most are interdependent. The variables include the type of support, the nature of the anchoring bond and how it is created, the method of coupling, the nature of the amino terminus protector and the deprotecting reagent, and the method of final deprotection. There are three major types of target molecules: peptides, which implies a free carboxy terminus, protected peptides, which usually implies a free carboxy terminus, and peptide amides. Resins are either microporous gels or composites. The more common gel is a polystyrene, divinyl benzene copolymer, an alternative is the polar polydimethylacrylamide, and made popular are the polyethylene glycol polystyrene adducts. The linkers to which the peptides are bound are substituted benzyl alcohols for the synthesis of peptides and substituted benzylamines for the synthesis of amides. Protectors are either tert-butoxycarbonyl for the amino termini with benzyl or variants for the side chain functional groups or 9,4-enolmethoxycarbonyl for the amino termini with tert-butyl for the side chain functional groups. Bond formation is by use of carbodiamides or variations thereof, occasionally symmetrical anhydrides or activated esters, and onium salt-based reagents. Each coupling method has its attractive features and limiting implications. Some residues such as asparaginol and glutaminol may require special consideration. Selection among the options is often influenced by the experience of the operator and the traditions of the laboratory. If you need peptides and related services, please don't forget to contact us.